As Pacey mentioned, my name is Sarah McCann. I lead the Camerades group at the Quest Centre for Responsible Research at the Berlin Institute of Health. Camerades stands for Collaborative Approach to Meta-Analysis and Review of Animal Data from Experimental Studies. And as the name suggests, we do research involving systematic reviews of preclinical studies, particularly animal studies relevant to human health. So today I'm going to talk about preclinical systematic review and meta-analysis as methodologies, and I'm going to use one of our published reviews in stroke research as an example. So a systematic review is a robust method to objectively, transparently, and reproducibly synthesize research evidence to inform decision-making. So the goal is to really find all of the literature available on a research question so that you can have as complete and unbiased view of the current knowledge base in a research area as possible. A systematic review can involve a qualitative synthesis of the literature, and it might also include a quantitative synthesis, which is called a meta-analysis. And this is a statistical method to quantitatively summarize results from individual studies. So a systematic review can include a meta-analysis, but it doesn't need to include a meta-analysis. And meta-analysis is distinct from meta-research in general. Some meta-analyses are done in the context of meta-research and some are not. Typically, a systematic review follows a structured process, which involves defining the research question, writing a protocol outlining the rationale, hypothesis, and planned methods of the review, conducting a systematic search across multiple databases to identify all potentially relevant studies, selecting studies to include in the review based on predefined inclusion and exclusion criteria, assessing the quality and risks of bias of the included studies, extracting all relevant information and data from the included studies, conducting a qualitative synthesis or a statistical meta-analysis combining the data, and finally reporting the systematic review in a transparent way for publication. If we look at a very simplified version of the research evidence pipeline, where preclinical research in cells or animals is used to identify and test potential treatments, promising findings are then advanced to clinical testing in humans, and successful treatments are then translated into clinical practice and guidelines. We know that systematic review occurs routinely in the translation of findings from clinical research to clinical practice. But there's often a translational gap between preclinical research and clinical research. And this is where the results of animal studies are not predictive of what happens in humans. So a treatment might be effective in treating stroke in animal models, but then fail to show any benefit for human stroke patients. And systematic review at the preclinical stage can help us understand what's going on here and inform the translation to clinical testing. In particular, the results of systematic reviews can help to guide and improve future preclinical research by helping to identify knowledge gaps and making sure future studies are fully justified, assessing the quality and range of existing evidence, estimating reporting biases, and informing 3Rs decisions. And these 3Rs are our replacement, reduction, and refinement of animal use in research. Preclinical systematic reviews can also help on the translation side to help explain discrepancies in preclinical versus clinical trial results, inform translation readiness, and inform clinical trial design. And in stroke research, translational failure is particularly well recognized, and systematic review has been used quite extensively to investigate the reasons for this failure and to help inform improvement strategies. And the paper that I'm going to discuss today is one of these reviews that looked at a promising treatment for stroke. It's a systematic review and meta-analysis of the efficacy of interleukin-1 receptor antagonist in animal models of stroke. And this was published in 2016. And we can see from the title that it's actually an update of a systematic review. So the original review was published in 2009, and this was a research area with a lot of activity. So we decided to do an update, and to our knowledge, this is the first update of a preclinical systematic review. Both the initial review and the update synthesized the evidence relating to the efficacy of interleukin-1 receptor antagonist, or IL-1RA, which it's known by, in animal models of stroke. And IL-1RA is an anti-inflammatory protein that's used clinically to treat rheumatoid arthritis, and it's considered to be a promising treatment for stroke. And the original systematic review from 2009 suggested substantial efficacy of IL-1RA, but identified a number of shortcomings. These included that the range of conditions under which efficacy was tested was narrow. The study quality was modest when scored against established checklists. For example, the methods weren't clearly described or measures to reduce bias weren't taken. And there was evidence consistent with a substantial publication bias in the literature. So in 2016, IL-1RA remained a promising treatment for stroke and additional preclinical evidence had been generated. So we set out to update our existing systematic review, as well as providing a summary of the current data for efficacy in the field. This was the first opportunity that we had to chart changes over time in a field and the possible impacts that systematic review had on the directions taken by investigators in the field. So our systematic review update followed the same methodological steps as our original systematic review, and these are standard steps in systematic review that I discussed earlier. I'm going to go through these in more detail now, using our study as an example of the approaches to the methodology and some of the results that you might see. And I'm going to explain the importance of the design considerations at each step and best practices and some of the pitfalls to avoid. So the first step in a systematic review is to formulate a research question, and a well-constructed research question is really essential for a systematic review. It should be clear, structured, and unambiguous. It should provide a meaningful answer. The question can be broad or narrow in focus, but it should be feasible and manageable for your research team to answer. Most research questions can be broken down into components using a question format tool. And this is helpful to construct a clear and concise research question, and also helps identify key concepts to include in a search strategy. So the most common question structure is the PICO format, or the PICO, for intervention studies. And this can be used for both clinical and preclinical research questions. Using PICO, we define the P for the population under study. And for preclinical reviews, this includes the animal disease model population. The I is for the intervention, the C for the comparison, and the O for the outcome measure of interest. So our research question in the PICO format is, what is the effect of IL-1RA compared to vehicle or no treatment in infarct volume in animal models of ischemic stroke? So our population is animal models of ischemic stroke. Our intervention is IL-1RA. Our comparator is vehicle or no treatment in the control group. And our outcome of interest is infarct volume, or how much brain damage occurred after stroke. The PICO for intervention reviews is the most common research question structure, but there are many other research question structures for different types of review questions. These are just a few examples. For example, COCO-POP for systematic reviews of incidence or prevalence, or PCC for concept analysis. So once we have a research question, we develop a protocol for our systematic review. And this is arguably one of the most important documents in the systematic review. And it lays out the detailed plan for the rationale for every step in the review. So it outlines the definition of the problem and the intervention under investigation, the specific research question the authors are trying to answer, the criteria for which studies will be eligible or assessed for inclusion in the review, the process for identifying, assessing, and summarizing the studies in the review, and how the systematic review process will be managed. And this is really important for a number of reasons, including to increase transparency and reproducibility, to minimize bias, and avoid duplication and waste. The protocol should be prepared before the review started and used as a guide to carry out the review. And there are three main ways to make a protocol available or public before starting a systematic review. You can register the protocol on a registry. This is the preferred method, and it can be done on a specific registry like Prospero or on the Open Science Framework. You can publish a protocol as a journal publication, or lastly, just make the protocol publicly available, ideally giving it a persistent identifier so that people can find it and it enables citation. So our protocol was published on our website at the beginning of our review. We then move on to developing the methods that we're going to use to select studies to include in the systematic review. And this step and all of the following steps that I'm going to discuss should be outlined in the protocol before the review starts. And the eligibility criteria set the boundaries for the review. So these criteria are usually set before the search strategy is developed, or maybe after a scoping or preliminary search has been carried out. So this can be a kind of an iterative process. The eligibility criteria are applied during citation screening to guide reviewers whether to include or exclude a study from the review. And the inclusion criteria define the key features a study must have to be included in a review, and the exclusion criteria define the factors that make a study ineligible for inclusion. And these criteria should cover things like the type of study design, the type of animal and population, the type of intervention and outcome measures that you're looking for. This is just an example of some of the inclusion and exclusion criteria from our review, related up here to the population, so the P from our PICO, and the intervention, the I from our PICO. We can see from the population that we included whole live animal models of ischemic occlusive stroke of the middle cerebral and anterior cerebral arteries or their branches, and we excluded other models of stroke, so hemorrhagic stroke and global ischemia, which are different forms of stroke, and we also excluded in vitro studies, so studies that weren't done in whole animals. And all of this information comes from our protocol. And the next step is to develop a search strategy to look for studies to include in the review. And this usually includes searching electronic databases like PubMed or Embase. And a systematic review should generally include at least 2 electronic databases because different databases index different journals. And we perform searches to identify stroke studies in animal models that tested IL-1 array. These were the search terms included in our search, and you can see that they include search terms related to IL-1 array, plus terms related to stroke, plus an animal filter to filter out, for example, clinical studies in patients, which we're not looking for. So the search is really critical. It's really the backbone of a systematic review. So remember that the aim of a systematic review is to really capture all relevant studies. And if the search strategy isn't comprehensive, there's a risk that relevant studies won't be found. And then the results or the conclusions of the review might be incomplete or even biased. So it's advisable to talk to a librarian or an information specialist for assistance with this step. There have been systematic reviews where it's been picked up by peer reviewers that there are studies missing from the review. And this then highlighted the fact that the search was not comprehensive enough and the review process had to start again from the beginning, which is obviously not an ideal position to be in once you've written up your review and submitted it for publication. So this is a really foundational step for a review. We then have at least 2 independent reviewers screen the records that are returned by the systematic searchers, and they screen them by title and abstract, first of all. We then have 2 independent reviewers assess each study at the full text stage against the pre-specified inclusion criteria, and at this stage, discrepancies between the 2 reviewers are resolved through discussion or consultation with a 3rd reviewer. Having these 2 independent reviewers at both of these steps is best practice to avoid errors or reduce errors and bias in the review. And it's important to note that reviewers receive comprehensive training and screening and assessing eligibility for inclusion to increase inter-rater reliability and reduce the likelihood of errors. Once we've completed this process, we have the set of included studies for our review. So this is a Prisma flow diagram from our review, and something that looks like this should be included with every published systematic review. This diagram visually summarizes the screening process. So it initially records the number of articles found, and then it makes the selection process transparent by reporting on decisions made at various stages of the systematic review. So the number of articles are recorded, starting with the number retrieved from each of the different databases. Up top here, we can see we retrieved 595 records in total. After we remove duplicates, because there's overlap in the journals that these databases index. So after we remove duplicates, we screened 434 records by title and abstract, excluded 405, and then we screened the full text of 29 articles. And you can see here the list of exclusion reasons for the articles that were then excluded at the full text stage. We included 11 studies from our review update, and we combined these with 14 studies from the original review to have a total of 25 studies in this systematic meta-analysis. So once we have our included studies, we want to extract predefined study characteristics and data from each included study. We then want to do a quality assessment of each included study. And in this review, we used a 15-item quality checklist that included things like whether the authors randomly assigned animals to group to reduce selection bias, or whether they blinded outcome assessment to reduce detection bias. We also assessed the range of evidence available against a 13-item checklist, which was generated by the Stroke Therapy Academic Industry Roundtable, or STARE. And this is a panel of experts in the field who developed these criteria to try and help guide more successful translation in the stroke field. So this is obviously like a field-specific list. And we have 2 independent reviewers, again, carrying out each of these steps for every included studies. Discrepancies at this stage were resolved through discussion and where necessary, consultation with a 3rd reviewer. Like in the study selection phase, reviewers are trained in data extraction and quality assessment to increase inter-rater reliability and reduce errors. Reporting in animal experiments is often really poor or inconsistent. And even when you have 2 experienced reviewers, which we had in this particular case, we still had discrepancies where the reviewers disagreed, and we want to reduce this as much as possible in the review. So this is an example of some of the study characteristics that we extracted and what a study characteristics table might look like if you're reading a published review. Here we have each included study on a separate line, and we've recorded study design elements like the animal species, the dose and time of administration of IL-1RA, and the anesthetic used during stroke induction. These are some of the results from the study quality assessment. So this shows the proportion of papers reporting each study quality measure along the x-axis here. And these numbers are split up by whether the studies were included in the original review in 2009, which is shown in blue, or in the update in 2016, which is shown in pink. And we can see that across the board, there was an increase in reporting study quality measures after the first review. For example, we can see in reporting random allocation to group, there are about 5% reporting this in the 2009 cohort of papers and almost 90% in 2016. And this table shows our assessment of the range of evidence available across the data set in 2009 and 2016, again split up. In 2016, there were 3 new criteria, so 11 out of 13 versus 8 out of 13 in 2009. These included testing the drug with animals with comorbidities, measuring the outcome at more remote time points, and looking at interaction studies with medications that are commonly used in stroke patients. So these are all important things to think about when translating the evidence. So that was our kind of qualitative synthesis of the results. And now we move on to a quantitative synthesis or meta-analysis. So remember, meta-analysis is just the statistical pooling of all the quantitative data from our studies. So how effective is IL-1RA compared to control when we look across the whole data set? So we pre-specified our statistical analysis plan in our protocol. We calculated effect sizes for each study. So an effect size is just the difference in infarct size between the IL-1RA treated group and the control group. And then we combined these effect sizes using meta-analytic models. And because preclinical studies are really heterogeneous, for example, they might use different animal species or strains or different methods to induce stroke, we expect there to be differences in the effect size reported by different studies. And this between study variability or between study differences is called heterogeneity. And a really important part of meta-analysis, especially in preclinical systematic reviews, is to investigate possible sources of this heterogeneity. And we did this using a technique called meta-regression. We also looked for the presence of possible reporting biases using funnel plot-based methods. So I'll go through a bit now about what all these methods mean by showing some examples of results from the paper. This is what's known as a timber plot. So, sorry, it's a bit small, but each of these dots or triangles on the graph represents an effect size from an individual experiment comparing IL-1RA to a control group. And the vertical lines through the dots represent the 95% confidence interval of each effect estimate. So this effect measure in particular shows the percentage reduction of infarct volume in the treated group compared to the control group. So we can see here that the effects range from over -50%, which means there was an increase in infarct volume in the treatment group, right up to over a 50% reduction in infarct volume with treatment here on the right-hand side. So the pooled meta-analytic estimate, when we combine all these studies, is shown in this grey bar in the background. And this spans the 95% confidence interval of our pooled effect. So overall, IL-1RA treatment reduced infarct volume by 36.2% compared to control. But we can see in this graph that there's quite a lot of variability between these effect sizes or between these studies. And this, as I said, is one of the most important parts of a preclinical systematic review in particular is to look for sources of this variability or sources of this heterogeneity. And this is probably more important than this overall effect. So one of the ways that we did this and the way that we did it is using meta-regression. This graph on the left-hand side looks at the route of administration used in the studies, which is a significant source of heterogeneity in this data set. So this is how the IL-1RA was delivered to the animals. Again, this grey bar in the background is the overall estimate of effect. So when all the studies are combined, this is our 36% and 95% confidence intervals. And here we've split up the data according to which route of administration they used. And the highest effect or the largest reduction in infarct volume is seen with intracerebroventricular administration. This is just injecting the drug straight into the brain. This is obviously not a route that we would prefer to use in humans as it's very invasive, but luckily we see here that the other routes, intravenous administration and subcutaneous administration, which are a lot less invasive, are also effective in this data set. So these are more likely to be useful from a translational perspective. And we see here on the right-hand graph that higher doses of IL-1RA are more effective in animals than the lower doses. And this is the type of information that can be useful for informing clinical trial design. So next, we looked for any indication of reporting bias in the literature included in our review. And this is important because systematic reviews can only include publicly available data or data that are made available to the review team. If there are unpublished data out there somewhere and these unpublished data differ systematically from the published data, then our meta-analysis, our understanding and our subsequent decision-making will be biased. So potentially missing data pose a threat to the validity of systematic reviews. And there are several types of reporting biases. We think that the most important ones in preclinical literature are outcome reporting bias and publication bias. Outcome reporting bias is the selective reporting of some outcomes, but not others, depending on the nature and direction of the results. For example, this might mean that a number of outcomes are measured in an experiment, but only the outcomes with a statistically significant difference between the treatment and control group are reported, and those with non-significant differences are not reported. And publication bias is the publication or non-publication of research findings depending on the nature and direction of the results. This usually means that neutral and negative studies take longer to publish or they remain unpublished altogether. And this means that they're less likely to be identified in systematic reviews. And then this leads to overstatement of efficacy in our meta-analysis. So it's not possible to test for reporting biases directly, but we can use what's known as a funnel plot as an indicator of possible reporting biases. So in this graph, the effect size of each study is plotted against disposition or variability. And each of the black dots represents an experiment included in our review. So the larger or more precise studies at the top of the graph here cluster around the pooled effect, while the smaller or less precise studies are scattered more widely at the bottom of the graph. And that gives us this kind of inverted funnel shape typical of these graphs. So the solid vertical line here is our pooled estimate. So that's our 36% efficacy overall. And in the absence of bias, this funnel should be symmetrical. But what we can see here is asymmetry, where we're missing black dots on the lower left-hand side of the graph. So these would be studies with a small or negative effect size and low precision. And these are the types of results that we would expect to be missing from a data set if there was reporting bias present. So that's where they are. So what we've done here is to fill in these theoretically missing studies with a technique called trim and fill, and that's what the white dots are. So we're trying to make this funnel symmetrical. We can then recalculate the overall effect, including these theoretically missing studies, and get an adjusted estimate of effect, which we can see here with the dotted line. So you can see that the dotted line is pulled to the left, it's pulled towards the zero line. So if these studies truly are missing, then we might be overestimating our effect. And this second graph tests for this asymmetry statistically using a method called Eggers regression. And both of these results indicate that there may be studies missing, which could potentially reduce the effect size that we've measured based on the included studies. So the main findings are that the quality and range of evidence improved in studies after the initial review. And we know that the authors of the original studies read the initial review and made changes based on the results. There was high heterogeneity that was partially explained by the variables route and dose of IL-1RA administration. And there was still evidence of possible reporting bias and inflated overall effect estimate. So some of the weaknesses or limitations of this study, our protocol was not registered. It was made public on our website before the start of the review, but it doesn't have a persistent digital identifier or registration number. We didn't assess risk of bias in our review. So we assessed a broad range of study quality items, including some related to risks of bias, like randomization and blinding, but we didn't do a formal risk of bias assessment. So we just analyzed reporting of these items. So there is a tool to assess risk of bias in animal studies. It's called the Circle Risk of Bias tool. It's based on the clinical risk of bias tool for clinical systematic reviews. And you can use this tool and generate results like these, showing the proportion of studies in your review at low risk of bias in green, unclear in yellow, or at high risk of bias in red. Further weaknesses include that there are now more advanced meta-analytic methods optimized for preclinical data than we used in this study. And we did not share any data or analysis code for this study, which is something we now do routinely, which just seems really important. So in summary, of good quality design and reporting for preclinical systematic reviews, it's really important to include the components of the PICO in the research question and the inclusion criteria for the review. It's really important that the protocol is pre-specified and ideally publicly registered before the start of the review. As I mentioned, the search strategy is really comprehensive, so you're really not missing any studies that might change the conclusions, the results of your review. that the study selection, so that initial title and abstract screening and the full text testing for eligibility is performed in duplicate by at least 2 independent reviewers. You provide a list of excluded studies with the reasons why studies were excluded from the review, and you see that in the PRISMA diagram. That you use a satisfactory method for assessing risk of bias, like a validated tool like the Circle Risk Bias tool for preclinical studies, or there are other tools for assessing risk of bias in other types of systematic reviews. That the data extraction is also performed in duplicate with a method for reconciling any differences between reviewers. That the studies are described in adequate detail in the publication using, for example, a study characteristics table. That the sources of funding for the included studies are recorded. So this is something that we did do. I didn't present it, but it's important to know if there might be any potential conflicts of interest in your included studies. And this can be especially important when studies are investigating interventions for stroke that might lead to pharmaceutical development, for example. When we're thinking about the synthesis for meta-analysis in particular, that appropriate statistical methods are used. That you assess the impact of any risks of bias in the data set, that you assess any heterogeneity, so try and work out where any heterogeneity is coming from in the data set, assess potential reporting biases, and report any potential conflicts of interest in the review team. So are there any conflicts or funding conflicts that you need to report that might influence how you conduct the systematic review? And we're going to look at some limitations of systematic reviews. We obviously think they're great. We do them all the time, but there are limitations. So meta-research has shown that the publication of systematic reviews has grown exponentially in recent years. Unfortunately, many of these reviews are unnecessary, misleading, and conflicting, or just really poor quality. So they often seem to serve as easily publishable units or marketing tools for researchers, which is not really what we want. And this is why it's really important to know what makes a good quality review, and when we conduct a review, that the research question is relevant to stakeholders that we're trying to address and addresses a valid gap in the literature. Systematic reviews require a lot of time and resources to conduct. A common criticism or limitation of systematic review is that they're rapidly outdated. This is especially true in preclinical reviews that can include up to hundreds of individual studies that need to be extracted and assessed. The measures of rigor that we have are indirect, so we can only rely on what the authors report. We don't know what they actually did in their study. So they might not have reported that they randomized, but they might have actually done it, or vice versa. We can only go on what they say they've done in the paper. The systematic review is only as good as the data that go in. So sometimes the phrase garbage in, garbage out is used. And this is why it's vital to conduct that critical appraisal of the included studies and use the results of this critical appraisal to inform your summary and synthesis and the conclusions of the systematic review and meta-analysis. And lastly, there are some tools to help us identify a high quality systematic review and high quality reporting. So there are reporting guidelines which outline all of the elements that should be included in a published systematic review. I mentioned the PRISMA flow diagram earlier. PRISMA stands for Preferred Reporting Items for Systematic Reviews and Meta-Analyses. These are general systematic review and meta-analysis guidelines. There are currently guidelines specific to preclinical systematic reviews under development, but at the moment we just use the general guidelines. There are also critical appraisal tools for systematic reviews. These include a measurement tool to assess systematic reviews, or AMSTAR-2 as it's known. This is a 16-item checklist that helps us to assess how the systematic review was planned and conducted and the quality of the systematic review methods. So the items that I have on the summary slide of good quality design and reporting included most of the items from AMSTAR-2. And that's all I have for this. So thank you very much for your attention and happy to take any questions.